I'm Karen Conneely. I'm in the Department of Human Genetics at Emory University. I was trained as a statistical geneticist, and then I became very interested in epigenetics. So now I guess I'm also a statistical epigeneticist. I have a particular interest in the epigenetics of aging, but that's not what I'm going to talk about today. Today I'm going to be talking to you about the analysis of whole genome bisulfite sequencing data. This has been billed as a research talk, which I think it is because part of the research I'm going to talk about is the stuff I was involved in, but not all of it. And so I think it is also a tutorial. And because my goal is to give you kind of a broad overview of the field. So we're going to start by giving an overview of DNA methylation, because I'm pretty sure I'm the first person to talk about this in this short course. So we're going to talk about what is DNA methylation and what does it do, how we measure it. And then we're going to focus on bisulfite sequencing data. We're going to talk about some specific challenges in the analysis of these data. In particular, we'll talk about how it's challenging in terms of alignment, and then we'll talk about differential methylation analysis. And so, oh shoot, I forgot I'm supposed to use this. Okay. So, as promised, what is DNA methylation? First of all, how many of you have already worked with DNA methylation data? So some of you, less than half. How many people, this is just completely new to you? So some of you, it's new. Okay, so I'll try to go through this, not too slow, but not too fast. DNA methylation is the addition of a methyl group to the 5 position of cytosine. It's considered an epigenetic modification, which is an external change that influences gene expression but does not alter the DNA sequence. And so this is the chemical addition of a methyl group to a cytosine. It primarily happens at cytosines that are followed by guanines, also known as CpG sites. And let's see if I can make the mouse work. There it goes. Okay. So does it work like this? No, I have to go like this. Okay, so I'll get the hang of this. Okay, so here we have a strand of DNA with some CGs on it, and there are methyl groups attached to each of the CGs, so they are methylated. Okay, so the next question is, why do we really care about DNA methylation? The answer is that it has a potential regulatory role. It's one of the epigenetic mechanisms that contributes to the regulation of gene expression. And so in humans, we have 28 million CpG sites across the genome. And most of these are methylated most of the time. And so the exceptions tend to be the interesting regulatory regions. And so one exception is the CpG-rich regions that tend to occur in promoter regions. These are known as CpG islands. The CpGs that are in these CpG islands they tend to be they tend to be unmethylated. So here is a strand of DNA with 2 unmethylated CGs on it. And when they're unmethylated, they can, in a CpG island, they're left free to be transcribed. And so when they become methylated, this can often suppress expression of the genes or silence the genes. And so that's the kind of canonical way that we think about DNA methylation. But there are other regions as well with regulatory potential. One of these is enhancers. And methylation in enhancers can be thought of as a dimmer switch that controls the expression levels in different cell types. Methylation in gene bodies, different work has found different associations with expression. Some work has found primarily positive correlations between methylation in gene bodies and expression, as compared to the negative correlation we see in promoter regions. Other work has seen that there's both positive and negative correlations in gene bodies. Okay, so since we've been talking mainly about genetics, I'll contrast the genotype to the epigenotype. So unlike your genotype, which you're assigned at birth and kind of stays with you throughout life, your epigenotype is quite dynamic. The changes in DNA methylation play a crucial role during development, and they continue to change across the lifespan. They can also be influenced by the environment, especially environmental factors like smoking or chemical exposure. And so this makes DNA methylation a great candidate to possibly mediate the influences of both gene and environment on resulting phenotypes. Also, like so aside from the rare somatic mutation, your genotype tends to be pretty consistent in all the cells of your body, whereas the epigenotype is going to vary from cell to cell. So changes in DNA methylation are heritable during cell division. And this is actually how cells differentiate and establish their identity during development in cell tissue differentiation. Because of this role in cell and tissue differentiation, it becomes very important in these epigenetic studies which tissue do we sample. So in genetic studies, we always take blood because it's easy to get. In DNA methylation studies, we often also take blood because it's easy to get, but we really think hard about it first and think about the tissue that we wish we had. And so in addition to that, regardless of the sample that we choose, the methylation patterns are not going to be the same in all of the cells. So, so here's a very simplistic picture of a sample of 7 cells. And a single CpG site, 3 of the cells have methylation at the site and 4 of the cells don't. And so we're at every CpG site, the ways we measure methylation will allow us to estimate a proportion of methylation rather than just a 0 or a 1 value. So next I'm going to talk about a few of the approaches to measure DNA methylation in these samples. The approaches can be divided into 2 categories. So there's capture or enrichment-based approaches. So these will use things like antibodies or methyl binding proteins to capture the methylated DNA fragments, also known as enriching for them. And once we have all the methylated DNA together, we'll go ahead and sequence it or use a microarray. And that allows us to pinpoint where the methylation is occurring across the genome. The other category of approaches that we'll discuss today are the bisulfite conversion-based approaches. In these, we use bisulfite treatment to treat the DNA, and this converts unmethylated cytosines to thymines. It leaves the methylated cytosines alone. What we then have are things that look a lot like these CT SNPs. And so we can then either sequence or genotype the DNA, look at the CT SNPs and see whether we get Cs or Ts or some mix of both. And that allows us to assess the methylation at every site. Okay, so I'll go through several of these approaches and we'll talk about some of their strengths and weaknesses before moving on to just bisulfite sequencing. So the most commonly used approach is these Illumina arrays. So how many people in here have used those? Probably of the people who raised their hands for methylation, most of them have used these arrays. This is a very commonly used approach. I'll discuss the reasons why. So this approach, like some others, relies on bisulfite treatment of the DNA. And so here, the diagram on the right shows what happens during bisulfite treatment. The bisulfite conversion of the DNA fragments converts the unmethylated C to a uracil. And this is a C that's not part of a CG, so it's always going to be unmethylated. Over here, we have a CpG, and so it could be either methylated or unmethylated. If it is methylated, it won't be converted to anything. If it is an unmethylated C, it will be converted to a uracil. So here we have, it's almost like we have 2 different alleles. So I mentioned CT before, but now I'm talking about uracil. That's because during the PCR amplification, the uracils just get converted back to thymines. And that's how we end up with these CT SNP things. And so here we have a bisulfite-treated strand of DNA. We have a C or a T over here. And our next step with the Illumina arrays would be to put these bisulfite-treated fragments on an array, and then we can genotype the CpGs at many different sites. And the state-of-the-art Illumina array that everyone uses right now is called the methylation epic array. And it looks at nearly 1 million CpG sites across the genome, coming to about 20 to 50 sites for each gene. So some of the advantages of this array and the reasons why everybody uses it is it's very cost-effective. So depending on whether you do the sample prep in your own lab or whether you send it all to a core, it'll cost between usually $250 to $400 per sample. It's very simple to run and analyze. Almost every core has the capacity to do this. Many labs can go ahead and do the sample prep themselves without the need for additional equipment. You get great resolution. You get single CpG resolution for like 850,000 CpGs. And it's been compared to sequencing, and it seems to give precision that's equivalent to about 100x coverage. And so this is really great. For the almost 1 million CpGs we get, we really have nice deep coverage. This high precision, along with the high ends we can usually get because it's so affordable, means that we can use very simple, straightforward statistical methods to analyze these data because the law of large numbers kicks in. Plain old t-tests will work. Linear regression will work fine. People like to use mixed models. And so that's part of why this lecture is not going to be about microarrays. The analysis methods are already very established for them and quite simple. And also for these reasons, everyone has used this array. And so we have a single standard. There's no competitors, really. There's no Affymetrix is not in the game. So we have many groups all using the same array. This allows easy collaboration. There have been many large meta-analyses using the kinds of data generated with these microarrays. So those were the advantages. Some challenges with this kind of data. So we know that this is not getting all of the 28 million CpG sites. It's just getting a fraction of them, less than 1 million. The array was designed to focus on promoters and CPGs that are in promoters and enhancers, the sum and gene bodies as well. But there are many regions of interest that we may miss, and we're not going to get all of the CPGs. Another challenge is that if you're studying something other than humans, this may not be a good choice. The array was designed for humans. It's been used with primates. But if you're studying mice, then there is no array for you. And another limitation is this is a limitation of all bisulfite conversion-based methods. When you measure methylation using a bisulfite-based method, this isn't going to distinguish DNA methylation from another form of methylation known as DNA hydroxymethylation. And if you're analyzing a blood sample, this turns out to not be very important because hydroxymethylation is quite low in blood. But in other tissues like brain, where it's more prevalent, this is more of a concern. So another category of approaches, and this actually addresses that last point, in that it can distinguish methylation from other forms of methylation. These are the capture-based sequencing approaches. And so what these involve are capture of the fragments of DNA with methylation on them, followed by sequencing. And so here's a diagram of that. So the fragments of DNA up here, they're double-stranded now. Some of them are methylated, some are not. They get denatured. And then an antibody comes along, and it picks up all of just the fragments that have methylation on them. The others just go away. And so we're left with a bunch of methylated fragments. We can perform sequencing on them and look at where the reads pile up, and that tells us something about where the methylation is occurring on the genome. And so in the example I showed you, it was using an antibody, but there are other methods to capture the methylated DNA as well. So some advantages of this approach is it's also quite cost-effective, a little bit more than arrays, but not that much more. It could potentially assess the entire genome. It picks anywhere where there's methylation, it's going to potentially be captured. It can be used for any organism. You probably need a reference genome, but if you have a reference genome for your organism, you can completely use this method. And as I mentioned, it is specific to DNA methylation, or you can also use it to assess hydroxymethylation if that's the kind of thing you're into. So some challenges. The resolution is lower compared to the arrays. So unlike the single CPG resolution we get with the arrays, here we're looking at fragments, and all we can tell is, does the fragment have methylation on it or does it not? So we can measure methylation in these 100 to 200 base pair windows rather than the single CPG resolution. So the data gets a little bit fuzzier. And then some other challenges are that here you are assessing relative rather than absolute methylation. You can't compute a proportion the way you can for the arrays, because you're seeing where the methylated reads pile up, but you don't see unmethylated fragments. They just went away. You don't know where they were. There's no way you can compute a proportion. So you're really just trying to assess where the methylation is and where it isn't. And you can't even necessarily look at one area relative to another and say, well, there's more methylation here than here. The proportion must be higher. Because there are other biases. For example, the pull down by the antibody is going to vary with CPG density. And so these measures really are very relative. And this means that some quite sophisticated methods are required. And so I've cited one of these methods at the bottom of this slide. Nick Johnson, who's here at the course, is also very interested in this topic and is considering developing new methods. And so if you're interested in this, you can talk to Nick some more. That's all I'm going to say about capture-based sequencing, because today's talk is about bisulfite sequencing. OK, so this is another bisulfite-based method where we treat the fragmented DNA with bisulfite. The unmethylated cytosines will become Ts. The methylated cytosines are protected and will remain a C. And we just have to go in and sequence and look to see whether there is a C and a T, a C or a T at each C base or each CPG. So some advantages of the whole genome bisulfite sequencing. So we're back to having single CPG resolution, which is great. And not only that, but we potentially have it for the entire genome. So we could, if we had enough coverage, we could cover the whole genome. We could capture all 28 million CPG sites and get a nice single CPG resolution, proportion estimate of methylation at each one. That's great. It can be used for any organism. It's probably better if you have a reference genome. I think it's theoretically possible to do a de novo assembly with this, but you would need really great coverage, which we never have. Another advantage of it is that you can not only look at the single CPG resolution, but you can look at neighboring CPGs that are on reads. And so here's an example. These are pictures of like methylation haplotypes from certain reads. And so this one is like 4 CPGs in a row, methylated, and then 3 unmethylated ones. But in another cell or another sample, it looks like 2 unmethylated followed by 2 methylated. And so there is some work out there that tries to analyze the sort of different methylation patterns or haplotypes within reads. This is also something Nick's interested in if you're just looking for excuses to talk to Nick. Okay, so some challenges. So compared to the other two approaches that we talked about, whole-genome BS-seq is very expensive. Just to get some kind of reasonable coverage, you're going to need to spend between $1,500 and $2,000 a sample. It requires kind of a sophisticated pipeline, both for the lab and then for the analysis afterwards. So it's much more of an undertaking especially than the arrays. And because of these factors, the sample size is often quite small. And this means that these standard methods that worked great for arrays, like linear regression or t-tests, they're not going to be appropriate or powerful in this case. And this is what motivates methods development for bisulfite sequencing data. Another challenge is that because of the expense and because of various QC issues that come up, the coverage often ends up being quite low. And so in addition to accounting for small n, your analysis is going to need to account for possible low coverage and varying levels of precision due to low coverage among different CPG sites and samples. And so these last two challenges are the ones we're going to try to address in today's lecture. And then a final limitation of this is the limitation of bisulfite conversion I mentioned before, where we may be estimating both methylation and hydroxymethylation. And that's an issue for another day. We're not going to really talk about that more today, but that is a concern with the bisulfite-based approaches. Okay, so one more affordable alternative to the whole genome bisulfite sequencing is something known as reduced representation bisulfite sequencing. And here the goal is to narrow the focus, and not focus on the entire genome, but to narrow the focus to just the CPG-rich areas. And if we can do that, then maybe we can reduce the number of reads necessary to cover the whole. We don't have to cover the whole genome, so we can instead focus on having depth of coverage instead of breadth of coverage. And so get deep coverage of the promoter regions. And so the approach I'm going to describe below will interrogate about 1% of the genome, but it will capture between 5 and 10% of all the CPG sites. And so you're getting a little more bang for your buck here. And so the standard approach to enrich for these CPG-rich regions of the genome is to use a restriction enzyme that will cut at the CPG sites. And if you do this, it leaves fragments with CPGs at either end. And if you then do a size selection, make the fragments kind of small but not too small, you're going to end up enriching for the fragments that have a lot of CPG sites and tend to be in CPG islands. And this is how you get the high coverage of more CPG sites for less coverage of the whole genome. And so after this step, you would do the same steps as whole genome bisulfite sequencing, bisulfite treat these fragments you've made, amplify them, and sequence them, followed by alignment. Okay, so speaking of alignment, so both the RRBS method that I just mentioned and the whole genome bisulfite sequencing face very similar analytical challenges. And so the two challenges we're going to talk about today are how the alignment of the genome isn't completely straightforward and some approaches that you would need to use instead. And then we'll talk about how to efficiently use the information when we have this low coverage and low sample size, especially when you want to do a differential methylation analysis. Okay, so as far as alignment, the bisulfite conversion complicates the alignment in a couple of ways. The obvious way is that some of the C's have been turned into T's. And so they're not going to match to the reference genome. And so this is already a problem, but another perhaps less obvious problem is what happens during the bisulfite conversion. And so up here we have a forward and a reverse strand. And so let's denature these two strands, and then they're each bisulfite treated. And so starting here, the reverse strand is the complement of the forward strand. Then bisulfite treatment happens. And so on the forward strand, this C was methylated, it stayed a C. These two Cs were unmethylated, so they became U's, and then nothing else happened. Going to the reverse strand, so over here we have two unmethylated C's as well. On the forward strand, these were G's, so nothing happened to them. On the reverse strand, they are C's, and they're unmethylated, so they get converted to uracils. Suddenly, the two strands are not complementary anymore. We have two different strands. The next thing that happens is PCR amplification. So complements of each of these strands get produced, and we now have 4 different kinds of strands hanging around. And so this is obviously a lot more complicated than the usual alignment. We're not going to be able to use the usual aligner. It's just trying to naively align to the reference sequence would be a real problem. And so as I mentioned, there are these 2 problems. We have the 4 different strands, and then there's also going to be these C-T mismatches where some of the C's have been converted to T's. And since this happens to the unmethylated DNA, if you just sort of naively try to match without worrying about it, it means that you're methylated. If you had a methylated fragment, it would align quite well because it hasn't been changed, and the unmethylated fragments would not align at all. So you would get this bias in favor of methylated strands, and you would misestimate methylation. So a possible solution to these issues is something known as in silico bisulfite conversion. And in silico means we use our computers to do it. We all know this. So we would take our reads and our reference sample. We've got our computer program now. We go ahead and switch all the C's to T's, align it, and then just put it back the way it was. And that's basically what in silico bisulfite conversion is. That's what BISMARK uses. BISMARK is an aligner that is based on bowtie, but is designed to be used with methylation data. And so it does this whole in silico conversion thing. And this diagram shows you how. So, so up here you have your original genomic fragment. You have 2 methylated C's on it. And then here it is after the bisulfite treatment. You find this thing in your library. And here it gets a little more complicated because you don't know if this was the original DNA that got bisulfite treated or if this is a complement of it, like if this happened after PCR amplification. So if it was the original DNA that got treated, then you're going to want to switch all the C's to T's and then align to both the forward and reverse strand of the reference genome down here in red, where all the C's have been changed to T's. But if it turns out this was the complement that got produced during PCR, then the C's would actually be G's and the T's would be A's. And so you would need to do a second conversion where a second version of this, where you make all the G's A's, and then you align to 2 other versions of the reference genome where the G's have been changed to A's. And so you're doing this all over the genome, but there's basically 4 comparisons that you're doing all over the genome. So for each one, you're going to get a best alignment, you're going to get an alignment score, and then maybe the best score out of the 4 becomes your actual alignment. And then what you do after that is you go back to the original configuration where the C's are C's and T's are T's. You set it all back to the way it was. You place it where it's supposed to be on the reference sequence. And then you go ahead and you do the genotyping. So like here you have a C, that means you have methylation. Here you have 2 T's, so these are, and they were supposed to be C's, so they're unmethylated C's. Here you have T's that were just T's, and the reference genome tells you that. And so here you can see this was methylated, and here you can see that this was an unmethylated C. And so the program BISMARK will do this for you, and you'll end up with data that I'll show you in a minute. So that's one way of doing the alignment. A problem with the in silico approach is that by converting all the C's to T's, we've really reduced the sequence complexity to just 3 bases. And this makes the search space for possible alignments bigger than it needs to be. So another approach is that you could, so if we see this bisulfite read here, here's just a little fragment of it. So we know that this, if you see the C, that you know you have a methylated C. It couldn't be anything else. Well, unless it's the complement, but we won't worry about that. If you see T's, these could either be original T's or they could be unmethylated C's. And so what you could do is you could do this thing where it allows the T's can all be compared to both, they can all be aligned to either C's or T's. The C's can only align to other C's, but the T's can be aligned to C's or T's. And so that's what another approach called BS-map does. What BS-map does, so they go through and for a particular read in your library, they'll basically make all the possible configurations where the T's are changed to C's or not. So here there's 3 T's, and so there's 2 to the 3rd power different configurations that this could be. So this is your original read. These are the 8 different configurations of it, and it tries aligning all of them to the reference genome, sort of like what BISMARK did. And then it also has to do the same thing for the complement strand where the G's get turned into A's. And so both of these are pretty computationally intensive. It's gone back and forth as to which one takes more time to run. I'm not sure what the winner is these days because new versions have come out. But this does, I think there's more comparisons to be made, but it also reduces the search space. And so there are trade-offs. So both of these mappers have advantages. There have been some evaluations done of both of them, and they reported that BS-map seems to have a greater proportion of uniquely mapping reads, at least in the evaluation that was done. BISMARK is more likely to have the uniqueness of mapping be independent of methylation status. There seemed to be a slight bias with BS-map. And it has slightly greater coverage, at least in the evaluation. It's also, one of the papers also suggested it was more user-friendly for extracting data. And it does seem to be the more popular choice. And I think this is in part because it's based on the bowtie aligner, which everybody uses. And in addition to these 2, there are many other sequence aligners available for this kind of data. And I've cited a review at the bottom here. OK, and then another way to improve your sequence alignment is through QC. I'm not going to talk too much about QC, but this is what, so at the beginning of, once your sequencing is done, you get these FASTQ files, and they come with these sequencing quality scores called FRED scores. You can use these to identify, so the nucleotides towards the end of the read tend to be of lower quality. Quality often drops off towards the end of the sequence. And you can see that here, like over 28 is considered really good, over 20 is considered pretty good. And they do tend to be dropping off as you get to like 70 or 80, the quality drops off. And so it's generally recommended that these reads be trimmed based on the quality scores. And because low quality things will hurt your alignment, this actually ends up, even though you're doing some trimming, you end up with kind of more data. You get more aligned reads. And you can, after you've done this, you can assess this at the end with M-bias plots. And here's 3 different M-bias plots that are showing different patterns of bias. And so what these are plotting is the position of a base along the read by the estimated methylation level at that base. And methylation levels should be independent of position because of the random fragmentation, the position on read should mean nothing. So if you see something like this, it means that you didn't do enough trimming at the end. And this one, it looks like maybe slight amounts of trimming at both ends. This one, some trimming at the beginning would have helped. And so this is one part of standard QC. You would probably do it both before and after you did the alignment. And then you receive, after you do the alignment, you get some data. And so what do the data look like? And so here is what it looks like before another QC step that we're going to do. And so here's a little fragment of the genome. And so there's 5 CpGs. There's 5 places where, on the reference genome where we know there's a CG. And there's reads that align to the reference genome. And at each CG, you can actually see if it's methylated, indicated by a black circle, or if it's unmethylated, indicated by a white circle. And one thing you might notice about this figure is that there seem to be a lot of exact duplicate reads. And this is a little bit unlikely because the genome is so vast and we didn't get that many reads. We shouldn't get reads that are just completely overlapping, especially not a lot of them. And so when you see something like this is indicative that there's, what we're actually seeing is PCR duplicates. And so a common QC step is to actually go ahead and remove these PCR duplicates. And then you end up with something like this. And it looks way less exciting here. Coverage something got lower. But this seems to be necessary to remove. Otherwise, it would be biased by the things that got PCR amplified more. And so now you have reads that align along the genome like this. You can use a methylation extractor. And it'll go through, and for each CpG along the genome, it will go ahead and it will, so the data you get out is going to look kind of like what's on the top. And so for the first CpG, you really just had one read that overlapped with it, and it was methylated. And so you get 1 out of 1. The next one, you had 4 reads that overlapped it. 2 are methylated and 2 are unmethylated. So you have 2 methylated reads, 4 total reads, and so half of them are methylated. And so the resulting data are going to be in quite a simple format. They just look like this. So here's you'll get a long list of the CpG sites. You could actually get close to 28 million. And for each one, you'll get the total number of reads, which I've indicated by n here, and then the number of them that were methylated. And so the data look very simple, which makes it tempting to do a very simple analysis. And the goal of this, usually the goal of many of these studies is to do a differential methylation analysis. So the typical goal of a study would be to compare methylation levels between 2 conditions. It could be between 2 groups of experimental animals that receive different treatments. It could be between adjacent tumor and normal tissue in human samples. This is a very common study design. And an important question is whether the groups contain biological replicates. And by biological replicates, I mean more than just one in each condition. And so many of us are used to these very large N studies where they're in the thousands or the 10,000s. But many of the earlier whole genome bisulfite sequencing studies could have 1 tumor and 1 normal sample and compare them. And then even the ones that do have biological replicates, it may be a small number like 2 or 3 or 5 or 10. So these are not large N studies. Because of this, because people get this data back that looks somewhat simple, and even though there are many CpG sites, the N is not very large, it seems like this may motivate the use of very simple approaches, which I'm calling here ad hoc approaches. So these would be things like Fisher's exact test or t-test. Because we have data that it's a very approachable data set. It's simple, I can understand it. I know how to do a Fisher's test, I'll just throw it in the Fisher's test. And that could be useful for a first pass, but I'm going to show how these approaches lose information and talk about some other approaches. So if you did have just 1 sample per group, so no biological replicates, there are many tests you could do. You could do a Fisher or a chi-square test. You could do a logistic regression, which is basically a chi-square test with additional covariates. So here's an example of just a single CpG site, and we did sequencing for 2 samples, a tumor sample that had 44 reads with 73% methylated, and a normal sample that had just 12 reads, but 67% of them were methylated. So you can go ahead and calculate your odds ratio. It's actually a pretty moderate odds ratio. 1.33 is not bad, but we're treating the unit of observation as reads here, which may be cheating already. N is really 2, but we're kind of treating our effective sample size as 56. But even then, even with the cheating, the p is only 0.73. So we don't have a lot of power. With Fisher's exact test, similarly, if we did a chi-square logistic regression, we would get almost the same answer. And this is not completely inappropriate. You do have a bunch of reads. We threw them together and we did a Fisher's exact test, which is kind of what you do. We did treat the reads as the unit of observation, but it's not terrible. It becomes less simple when we have more than just one sample in each condition. So for Fisher's exact test, if we have biological replicates, we can't just do Fisher's test anymore. We would probably need to collapse the read information within groups. And so here's an example of a single CPG site that was sequenced for 4 different samples, 2 tumor and 2 normal. So now we have our original adjacent tumor and normal, which we can assume came from one person. And so we still have 32 out of 44 and 8 out of 12 methylated reads. And now we have a second sample, so like kind of a biological replicate. And so now we have 10 reads for the tumor, but only 4 are methylated, and 34 reads for the normal, adjacent normal, and 12 are methylated. And so if we wanted to do this in a Fisher table, we would have to kind of go in and add them together. So here we have 32 plus 4 makes 36, and so on. We could do this table. We could compute an odds ratio, and now we've got the odds ratio just doubled, and now we have a p-value that's a little bit significant. And this seems a little bit odd because the first sample is the same as before. It was 73% here and 67% here. The second sample is not that, the tumor and normal methylation is not that different either. We have 40% in the tumor and about 35% in the normal, which also doesn't seem like a huge difference. But our odds ratio just basically doubled. Does anyone see why? Does anyone see what happened here? Okay, so we didn't cheat. We didn't do it on purpose. But we ignored something that was really important. We ignored the fact that we had two different human beings contributing samples, and they were not the same person. They were not genetically identical. This person had 40% methylation in their tumor and 35% in their normal sample. This person was at 73 and 67. The two people clearly had quite different methylation levels to start with. And then we ended up with, for some reason, getting more, we just randomly got more reads for the first person's tumor. We got more reads for the second person's normal sample. So we were effectively comparing this person's tumor sample to this person's normal sample. When we added them together like this, these got weighted disproportionately from the other two. So this is a little bit like the classic problem of population stratification. If our cases came from one population and our controls came from another. That's kind of what we accidentally did here. And so what we did can also be termed as ignoring the problem of biological variation. So to do this test, we collapsed the read information across the samples. And this ignored any biological variation that might be going on between the samples. Biological variation is basically heterogeneity between the samples. It's natural, in this situation, it's natural variation in the underlying fraction of DNA between two samples in the same condition. And this is in contrast to technical variation, which is just the variation we get due to the fact that we're estimating the methylation levels. And we've sampled a limited number of reads from the larger pool of DNA. And here's a diagram to kind of help illustrate the differences between biological and technical variation. So here we have two, these could be considered replicate mice that are from the same biological condition. The mice might even be genetically identical, but they're still different mice. And so they're going to have different levels, they may have different levels of methylation just due to life experiences or randomness. So the differences between these two mice are considered biological variation. We then take samples from the two mice, and we're not taking a sample of their complete DNA. We're just getting partial, we're getting some of their DNA, and then we lose reads due to for various reasons. And so we do a lot of sampling, and the variation, so if we took multiple samples from the same mouse, we would get different estimates of methylation. And so that part is what's known as technical variation. And so when we collapsed into that Fisher table, we were essentially assuming that, because we were assuming that the samples within the group had that they had the same underlying fraction of DNA methylated, that means we went and attributed all the variation to technical variation, which was probably not correct. So let's try another ad hoc approach. So another approach would be the t-test. Here we have the same CPG site for the same 4 samples. So we've got the same read counts in the same proportions as before. We have 73%, 67%, and then 40 and 35%. Now we're just going to throw these proportions that we've computed into a t-test. And we can look at the mean proportions for tumor versus normal. There's a very small difference. It is not significant. And so now we got a very different result. We got a significant result with the Fisher test. We got a very insignificant result with the t-test. And here we did account for the heterogeneity between the samples. We used the proportions, and the proportions were different. So we treated each sample as independent of each other. So we did correctly account for the biological variation. But now we didn't account for the technical variation. When we computed the proportions and then threw them into a t-test, we were inherently giving equal weight to each sample. And this did not account for the technical variation due to some samples having much greater read depth than others. And so this is something that we needed to account for. We didn't. It would be possible to establish a weighted t-test or a weighted regression that would take care of this. But another issue that we saw with this approach is the very small number of samples. Here, our effective number of samples was 4, and we had low power, and we suffered from it. So we clearly need to have better approaches than these. We need approaches that account for both biological and technical variation. And so the challenges here were the limited information available at each site due to both the low number of samples and the low number of reads. The approach I talk about next is going to address this problem by borrowing information across sites, and is also going to account for both the biological and the technical variation. And so this Bayesian hierarchical model that we're about to talk about, so this was the work of Harry Feng. He was just a master's student when he began this work. He's now beginning an assistant professor job at Case Western, so he has come a long way. So Harry's method is going to account for both the technical and biological variation, as we've talked about. And then it's going to use an empirical Bayesian shrinkage approach that is going to borrow information about variation across the genome. And the reason to do this is because we expect to have a low number of samples. Information per CpG site is very limited in this scenario. It becomes very difficult to estimate certain quantities, especially things like variance and dispersion. And so first I'll talk about how Harry modeled the technical and the biological variation. And so with the technical variation, it's actually very straightforward to model. So we can assume that there is a true methylation for CpG I, biological condition J, and individual K. We will assume that there is a true underlying methylation proportion that we can't see, and we'll call it pi ijk. Instead of, we can't observe pi. Instead, we observe the number of reads that cover CpGI, which we'll call nijk, and then the number of reads that are methylated, which we'll call mij. So we actually know that M is binomially distributed. We sampled n reads. The true proportion of reads that should have been methylated is pi. So the number that are methylated are going to be distributed binomial with parameters n and pi. This actually turns out to be this is the exact distribution for that sort of thing. It's not an assumed distribution, kind of the way Fisher's test is Fisher's exact test is an exact test. And by doing this, by modeling this with a binomial distribution, we've now accounted for the differences in variation occurring because the samples have different numbers of read counts. So modeling the biological variation isn't quite as straightforward. What we need to do here is allow the pi parameter to be different across individuals, even individuals in the same condition. And we need to do that by assuming a distribution for pi. There's not going to be an exact distribution for pi the way there was for m. But it turns out to be reasonable and convenient to model this as a beta distribution. So the beta distribution is a 2-parameter distribution. On the right here are some pictures of beta distributions. Well, on the right over there, there's some pictures of beta distributions. You can see there's a whole bunch of different ones. It includes the uniform distribution. Even though it's only 2 parameters, it's very flexible, and you can have a whole range of them. This is one of the things that I think makes it reasonable. Another is that it's designed specifically to it only models variables that fall between 0 and 1. So it is a good choice to model proportions. And then the reason it's so convenient is because it's the conjugate prior for the binomial distribution. And for those who haven't heard this term before, this just means that it plays well with the binomial distribution. And here I'll show you how. So we're essentially doing a beta binomial mixture distribution. We have a conditional distribution of the number m, the number of methylated reads, conditional on pi and n. And then we give pi a distribution of its own that's beta. And if we combine them together, that's a mixture distribution. And we could get the joint distribution of m and pi just by multiplying the red and the blue distribution together like this. And then if we integrate it out over pi, this would give us the sort of marginal distribution of m. It's really conditional on n, but we don't care so much about n, so it's almost marginal. And that's actually our goal. We want to do that. And to do it, I've actually plugged the distributions in here. They're still in red and blue. This thing here, you might see this function that says b and then a comma, or b and then alpha of beta. This stopped working. I don't know why. Oh, there, it came back. There. OK, so there's a little b. So this is a beta function. And we're not going to really talk about that except to note that it's a function of the parameters, but not of pi. So it doesn't really matter. We can pull it out of the integral here, just like we pulled out the n and the m, because we're just trying to integrate out pi. We also have these pi terms, pi to the m over here, pi to the alpha minus 1 over here. We can combine them like this. We can combine the 1 minus pi term. And we've just simplified it. And this is what I mean by the beta and the binomial playing well together. They simplify nicely like this. And it actually makes the integration possible. Mixture distributions, it's not always possible to integrate out and get a closed form solution. But here we can, because if you look at this, it actually looks a lot like the beta distribution that we wrote out up here. It looks like just a different beta distribution with slightly different parameters. It's missing this beta term here, but this is really just a constant if we think of it in terms of pi. So we could multiply the constant back in if we wanted to. What we know is that if this were the PDF of a beta distribution and we integrated it over the full range of the beta distribution, we would get 1. If we got 1, that would be great. It would make all this stuff go away and we would have an answer. And so really this ends up, if we just plug in the right constants, multiply and divide by constants, this becomes 1, it falls out, and we get something that looks like this. It's not great. I mean, it still has these betas in it, and we haven't really talked about what beta is, the beta function is, and we're not going to. But it's a closed form solution. It's simple. You could compute it using R. You could compute it. You might be able to compute it by hand. It's the closed form solution is actually kind of a rare thing when you're dealing with all these mixture distributions and integrals. So this is a nice thing, and this is what a conjugate distribution basically means. OK, so I did this in terms of alpha and beta because it actually made the math less messy. But for our purposes, it's going to become convenient to reparameterize in terms of the mean and the variance of the beta distribution. So here we have the mean, and here we actually have a dispersion parameter, which can be thought of as a mean standardized variance. It's a variance that doesn't depend on the mean. And we're going to reparameterize our beta distribution in terms of mu and phi here. And that gives us our beta binomial hierarchical model. And so the idea here is that we have two groups, two conditions. Each one has different parameters. The biological replicates in each condition can be thought of as draws from the distribution. So the pi was the draw from the beta distribution, and then we draw reads from each replicate, and so those are drawn via the binomial distribution. And so the top part of this model is modeling the biological variation. The bottom part is modeling the technical variation. And our overall goal here is that we have these two distributions, and we want to test whether they're the same or different. And specifically, we want to test whether the mean is the same or different. Is there more methylation at this site in one group than in the other? So we can do this by testing whether the mu1 and mu2 are significantly different. And there are different ways to do this. You could try to maximize the likelihood. That turns out to be very kind of intractable and messy. There are different ways of doing it. An alternative approach would be to establish a wall test that is an equivalent test of this. And so here's the basics of the wall test. If you can get estimates of the mean, you can look at their difference, and then just divide by the square root of the variance. It looks almost like a t-test. We did have to derive the variance a little bit from the reparameterized beta binomial distribution we just made. And here, if you write it all out, its expectation is just mu, which is great. Means are easy. The variance isn't that bad. It's a function of the mean and of this dispersion parameter. And so if we want to go ahead and make this wall statistic, all we have to do is come up with nice estimates of the mean and the dispersion parameter, which we can do, right? And so that turns out to be one last challenge. And so I mentioned that we have this limitation due to the small number of samples, and it makes things like the dispersion parameter difficult to estimate. In particular, the estimates are just going to vary wildly if you have just 2 to 3 replicates per group. So our solution was to borrow information from CPG sites across the genome to help ourselves obtain reasonable estimates of phi. So to obtain the stable dispersion with just a few samples, we're going to impose a prior on the data. And here we're going to impose a log normal prior. I've just written this out. And you can see it's, so the PDF of phi is a function of, in addition to phi, it's got these two parameters. We're calling them M0 and R0. And we're actually going to estimate M0, instead of just assuming them, we're going to estimate M0 and R0 empirically from our data. We can get method of moments estimates from our actual data set. And that makes this empirical Bayes rather than a classical Bayesian approach where you might start by assuming a prior parameter as well. Our choice of the log normal prior was the one choice that we had to make here. This was motivated by empirical distributions from data that were available at the time. But our simulations, it turns out that it's robust to departures from this assumption. If we have other distributions as well, it doesn't seem to affect the estimation. The important thing is that we use is how this shrinkage approach works, which I'll try to show you. Am I like completely out of time? Yeah, I'll try to get through this fast. Okay. Okay, so we're going to estimate the phi by maximizing this posterior likelihood. And I'm going through the math really fast here. I'm not going to dwell on it, but it ends up looking, we end up maximizing this log function that looks like this. And we have these two terms at the end that are functions of these empirical things that we've estimated. And these actually turn out to be penalties. So they make our likelihood smaller if phi is too big or if the log of phi is too far from the distribution mean. And so what we're doing is penalizing phi for being too large or too far from its mean. And so that's what the prior is doing in the shrinkage approach. It's providing a referee that kind of keeps our estimates of the dispersion in bounds and keeps them from just kind of varying wildly all over the place. And so I'll just jump ahead to the results. So using this approach, the light blue is our full Bayesian hierarchical model with the shrinkage. The dark blue is without the shrinkage. And so you can see the shrinkage makes it do slightly better. Compared to the certain naive approaches we discussed earlier, it definitely does better in terms of the PPV. Interestingly, the difference is large if you have only 2 replicates per group, and it starts to shrink if you have as much as 5 replicates per group. And so it is true that the simpler ad hoc methods will start to work better when you have more numbers, just due to things like the central limit theorem and the law of large numbers. There's also been an independent evaluation done recently since we talked about that. And it looks like, so this method, which is implemented in DSS, performs well both in terms of ROC curves, in terms of having a nice, I've highlighted it here, in terms of having a conservative type 1 error rate, a nice uniform distribution of p-values like it's supposed to. And just to conclude, there are many strengths of BS-seq, as well as many challenges. What the strengths are, it's very ideal for detecting regions that are extremely dysregulated across biological conditions. And so here's an example from a cancer study where you compare tumor to normal. It's very common to see these large dysregulated regions where the blue is the normal. It's an unmethylated promoter region, and the cancer samples, its methylation is completely dysregulated. For other studies, BS-seq may not be the answer. So so I study human aging, where we're looking at very subtle changes that may occur over the lifespan. Here we may not, at the state we're at now, I don't think we have the coverage to pick up these kinds of changes with things like whole genome bisulfite sequencing, not with the coverage and the samples that we currently have access to. So in certain studies, arrays may still be the best solution. And so we do have, as I described at the beginning, we have several options for designing methylation studies. And so it's important to consider which one best fits your study, whether you need breadth of coverage versus depth of coverage, for example. And then we've talked about some of the challenges that are inherent to bisulfite sequencing data and ways to address them. And with that, I'm going to thank you for your attention. I apologize for going over. I want to acknowledge my collaborators. Hao and I co-advised Harry on the work in the latter part of this talk. And Harry did all the work, of course. This is my lab at Emory, at least the way it used to be before people started graduating, Chloe. And I might be the only person here who's not currently hiring a postdoc, but I do hope to be in maybe 6 to 8 months. So if you're interested in epigenetics, in particular the epigenetics of aging, please come talk to me and we'll keep in touch. And with that, I don't know if I have time for questions, but thank you again. Thanks.